Good afternoon to the respected teachers, my dear colleagues and my juniors. My topic of presentation is ECM Remodeling in Cancer. These are my contents to be delivered. So we will begin with the change in the approach which happened since the last 25 years. Formerly, we have been focusing on the dynamics a cell of the cancer undergoes in order to promote all its events like malignancy and all. But the approach is shifting in the sense that now we have started considering tumor as an entire organ, and in order to address its entirety, we have started using term tumor microenvironment, which has certain components like tumor vasculature, extracellular matrix, connective tissue, and infiltrating immune cells. Extracellular matrix may have further different components, out of which I have mentioned some like fibrillar collagens, elastin, proteoglycans, hyaluronic acid, fibronectins, and laminins. Things which have been highlighted in red will be discussed in detail. Too much is too disordered. Like the solid tumors are characterized by the high levels of various ECM molecules, which compromises or let me say kind of hijacks 60% of the tumor mass. The sources of these highly expressed ECM molecules are tumor cells themselves as well as cancer-associated fibroblasts. Here is a signaling lobe, a probable signaling lobe, which is presented in the simplified form. So you can see the cancer cells are signaling by the TGF beta pathway to the quiescent fibroblast cells, which are converted to the cancer-associated fibroblasts. These cancer-associated fibroblasts are further contributing to the downstream signaling through cytokines, matrix metalloproteinases, etc., and contribute to the ECM stiffness and cancer cell progression. Macrophages also contribute to the same pathway by converting into the M2 macrophages, which in turn progresses to the TGF beta pathway and converts fibroblasts to the CAF, that is the cancer-associated fibroblast. But as you saw in the last diagram, the cancer-associated fibroblast do not have a single origin. It may have different kinds of origin like pericytes, ciliate cells, tissue-resident fibroblasts, mesenchymal stem cells, fibrocytes, etc., and thus contribute in different kinds of the cancer-associated phenotypes. This is the presentation of normal ECM versus cancer ECM. So you can see on the left hand side normal ECM being clear in deposition, being so ordered in deposition, whereas with increasing matrix stiffness you can see the traffic inside the interstitium increasing manifold. So collagen deposition has been discovered in different cases of cancer as a marker of several cancer events like invasive ductal carcinoma. You can see the upregulation of collagen 1 and 3. In desmoplastic breast carcinoma, it is collagen 5 which is being upregulated, contributing to 15% of the total collagenous composition. Melanoma, collagen 1 being overregulated. Ovarian carcinoma, collagen 4 being downregulated, and human lung tumors, collagen 1 by collagen 3 ratio being upregulated and collagen 4 being downregulated. In this case, I would like to highlight collagen 4 downregulation is associated with the aberrant deposition in the basement membrane, which leads to the disruption of the basement membrane. The deposition, if we are talking in respect of collagen, the deposition is actually related with the background expression of different contributing enzymes like lysyl hydroxylase, prolyl hydroxylase, and lysyl oxidase, which are the markers of invasiveness and chemotherapeutic resistance in different cases of the cancer. If we focus on a dynamics of laminin, laminin is again in normal tissues exclusively expressed in the basement membrane, and when you stain laminin, then you can find the well delineated boundary. But in the case of the cancer, you can see the overexpressed laminin in the basement membrane as well as in the stromal area, and the distribution is aberrant. This is being shown in the diagram here. In the figure A and B, the normal breast tissues are being shown with positive laminin expression, whereas as you can see in figure C and D, you can see the expression is both positive and strong positive. So in the upper two pictures, you can see the sharp and fine delineation of the basement membrane, where in the below two pictures you can see kind of haziness and kind of aberrant distribution marking it very deep and dark. Epithelial mesenchymal transition, one of the very well-known, one of the very most discussed phenomenon in cancer, is characterized by the polarization of the epithelial cells into mobile and ECM components secreting mesenchymal cells. Diagram shown below is showing the progressive loss of the polarity of the ECM cells, which were formerly connected so well in the ECM, but after the epithelial mesenchymal transition, the markers change. For example, the E-cadherin, which is the marker of the normal tissue, is down-regulated in the modified cells, and the marker changes to bimantine or other like fibronectin. There are certain evidences which correlate EMT with matrix stiffness, like in a study conducted by Rice et al. The PDAC cells are plated on different substrates with different rigidity or stiffness, and as a result it has been seen that there is a nuclear localization of the transcription factor YAP and PAZ, and this leads to the epithelial mesenchymal transition, which is further characterized by the loss of E-cadherin markers, by the gain of bimantine markers, and resistance to drug like paclitaxel. This picture is a schematic representation of what I just spoke. You see at 1 kilopascal pressure on the substrate, less of the cells are showing this brown stained nucleus, whereas as you increase the pressure to 4 kilopascals, more of the nucleus are showing the brown stained nucleus, and as you increase the pressure to 25 kilopascal, almost all the cells are expressing the YAP and TAZ localization into the nucleus, which leads to the upregulation of the mesenchymal markers as well as the related phenotypes. Well, there are some other evidences associated with this, like stiff ECM drives EMT breast cancer cells by promoting twist one transpolarization into the nucleus. The same is what happening with the YAP and TAZ. In gastric cancer, the HA-HMNR axis is linked to the EMT and 5-2 resistance. The highly cross-linked collagenous matrix increases matrix stiffness and focal adhesion kinase mediators in the lead responsible for malignancy. If you see the correlation of the drug resistance with matrix stiffness, you can see here that the overexpression and the as a result traffic into the interstitium creates a mechanical barrier for the drugs which are provided to the patients and become the cause of the barrier. This also leads to fall in the concentration of oxygen and nutrition in the interstitium, and further it produces hypoxia-inducing factor, which is responsible for providing the cancer cells with different kinds of adaptations, and is also responsible for inducing the cancer cells with the certain transporters which are responsible for the efflux of the drugs taken up by the cancer cells. Another, it's not that the direct mechanical pressure from the ECM deposition is only possible factors for drug resistance. In pancreatic ductal adenocarcinoma, you can see the high hyaluronate content is indirectly promoting drug resistance by accumulating high amount of water, which in turn increases the interstitial fluid pressure, and that kind of compresses the blood vessels, due to which the diffusion of the drug throughout or extravasation of the molecules of the drug through the blood vessels is inhibited. If I discuss the efforts being made to undo this stiff matrix phenotype, the first is targeting LOX. LOX is lysyl oxidase, responsible for the cross-linking of molecules like elastin and collagen. So they have used, this is a kind of amino oxidase enzyme which oxidatively deaminates specific peptide lysine and hydroxylysine residues, which is essential for establishment of collagen networks. This affects the drug distribution. So what they have done for targeting LOX is using beta-amine, that is 2-aminopropionitrile, which is a reversible inhibitor of the molecule, and using this we have seen that it reverses the phenotype like invasiveness and matrix stiffness. Cancer-associated fibroblast may also be targeted, but there is a clause in this that you cannot randomly target any CAF, because they may be associated with different kinds of functions. Like CAFs are also involved in immune function. So many times it has been seen that targeting the CAF reduces the immune program of the individual so deeply that the person dies. So in order to do that, in order to tackle that, we have to understand the functions of individual CAFs because they are heterogeneous in origin. And one example is illustrated here, like the one CAF illustrating CD10 GPR77 plus marker is downregulated or is targeted by the antibody against GPR77, and that has increased the sensitivity to the cytotoxic treatment. Another approach which is being developed, which is still under development, is reprogramming CAFs, which is approach to turn back the differentiated CAF back to the quiescent stage, which I discussed in the beginning 2 slides. And for this, they are targeting nuclear factor kappa B signaling, which is one of the activating pathways for cancer-associated fibroblasts. And in doing so, they have seen that they have increased the resistance to cisplatin drug. The conclusion is that mammalian ECM contains about 300 proteins, and many of them overexpressed during cancers. Various signaling are activated as a result of these pathways. Aim of the researchers is that to target the problem by understanding different functions of cellular and non-cellular components. The subsequent correction of the ECM expression profile will definitely help in reversing the phenotype and making cancer cells more available for action of drugs. These are my references. Thank you so much.